So I'll probably firstly want to thank everybody for being here. I know it's been a long day, so hopefully you guys can get well rested on this Monday after this talk. But first I have to keep you awake, so let me try to do that. I also want to thank the organizers for having me here. It's a pleasure to be here at ISMB, and particularly in Montreal, it's quite nice. So I've been enjoying that. So today I actually wanted to talk about one aspect of network-based analysis in protein folding dynamics that our lab has been working on quite recently. And just to start, my talk will be outlined as such. So as we know, a protein's amino acid sequence will fold into a particular 3D structure or a 3D structure. And the shape of this 3D structure will actually indicate what kind of cellular roles it might take on or what kind of interactions it might have with other biomolecules. This is knowledge that's quite common to everyone. But we also know that analyses of these protein structures are quite critical for our understanding of how these proteins might have a particular cellular role. But since more and more proteins are being discovered by current sequencing technologies and wet lab experiments for determining these cellular roles are not really able to scale quite well, we don't know a lot about the cellular roles of many proteins. So computational methods that can predict these cellular roles are quite important. Now, traditionally, these computational methods will extract features from either the protein sequences or the 3D structures, and then use them for various downstream machine learning tasks and biological applications. But over the last decade, there's been a shift to model these 3D structures as protein structure networks, or for short PSN, where nodes in this network are amino acids, and an edge exists between two nodes if their corresponding amino acids are close in 3D fold. And because we're using a network representation, well, we can use a whole lot of different network methods to extract features from these PSNs. We and others have shown that PSN approaches are quite effective in the task of protein structure classification, particularly having higher accuracy and lower runtime. You can also see this kind of task as a form of validation, where given the features of a protein, whether it be sequence or 3D structural, one can correctly predict these features corresponding to which structural classes, such as those in cath and SCOPy. And moreover, it was concluded by these authors, if I can use my laser, these authors, that graph-based deep learning, specifically graph convolutional networks or GCNs, were superior to traditional non-network-based approaches that work directly on sequence and 3D structural data, particularly in the task of protein function prediction. And later on, because these traditional approaches for studying PSNs were based on graphlets or these Lego-like building blocks that are really good for summarizing networks, the same authors evaluated that graphlets and GCNs are on PSNs in particular, that these graphlets outperform actually this graph-based deep learning method. So at this point, hopefully I've shown that PSNs are an excellent network representation, particularly for protein structures, and hopefully that graphlets are a somewhat effective tool, or at least I believe a very effective tool, for extracting important features from these PSNs. And if you're not convinced, I certainly can talk to you about that after the talk, so because I'm particularly convinced. And hopefully I can convince you more in this talk. Now, it's important to note that these traditional PSN methods model the native 3D structure of a protein as a static PSN, meaning that they only captured the native structure of a protein as a static network. But we know that protein folding is not this static process, right? Rather, it's a temporal process, as illustrated here, where, if I can pull up my laser, cool, where the protein's amino acid structure or amino acid sequence folds gradually into larger and larger subsequences or substructures, also known as protein intermediates, until it reaches the final native structure of the protein. Now, ideally, our lab would want to capture this dynamic information, these protein substructures or these intermediates, in our PSN model, but there are very few kind of experimental methods that can actually obtain this dynamic data. More so, even the experimental data that's on protein 3D substructures are severely lacking. And so we're praying that some experimentalists might pick that up at some point. So as our lab had to compromise. to develop a proxy for mimicking this kind of protein folding process by constructing a dynamic PSN type using static data, namely the native structures of the proteins. Now, while both static and dynamic PSNs are constructed using a network's native structure, the static PSNs are only capturing the whole native structure as one static network, as illustrated here. Whereas dynamic PSNs actually capture growing subsequence and substructure via snapshots, where in the first snapshot, we're just capturing a small subsequence and substructure of the protein, and with more and more snapshots, we're grabbing larger and larger subsequences and substructures of the protein until we arrive at the final snapshot, which captures the native structure of our protein. Now, in light of this new PSN model, our lab asked, would modeling native 3D protein structures as dynamic rather than static PSNs improve protein structure classification performance? And to test this hypothesis, our lab designed such a methodology where we compared dynamic PSNs to static PSNs in, of course, the task of protein structural classification. And now, in particular, we did use these 3 PSN types today, and I won't go into detail in these 3, but if you'd like to discuss them later, I certainly can. But these are the 3 static PSN types that we compared to. Our lab used all 44,000 protein domains that had structures in the PDB, as well as had structural labels in the Cath and SCOPy database. And these 44,000 protein domains, which correspond to 44,000 PSNs, were spanned in about 72, or exactly 72 data sets, depending on the hierarchy that's in Cath and SCOPy. Cath and SCOPy. And because our lab worked with a network-based model, we can utilize a network-based approach to extract features, right? And we employ graphlets, and we extract corresponding graphlet features for each of the PSN types. So in this way, our comparison between the PSNs is actually fair, because any potential differences that might arise between these PSN types will actually be due just to the PSN characteristics rather than their features. Our lab produced these results where in red are the dynamic PSNs, and in the blue, gray, and black are the static PSN types. On the x-axis are the 72 data sets our lab tested on, and on the y-axis is the misclassification rate, where lower misclassification rate indicates better performance. Now, when comparing our dynamic PSNs to the other PSN types, our lab found that dynamic PSNs significantly outperformed the static PSN types in the majority of these data sets. So in other words, despite the dynamic PSN model only being constructed from the final static network or the final native structure of the protein, these dynamic PSNs are performing quite well in the task of protein structure classification. So from this previous work, let me talk about what motivated our lab's current work. First, I should recall what I said earlier, that experimental data on these protein 3D substructures are severely lacking, and we had to ultimately compromise by designing a dynamic PSN model using static data, right, these native 3D structures. Well, we ask, will capturing dynamic data rather than static data with dynamic PSNs yield more meaningful results, right? And thankfully, we did find some dynamic data, namely protein structural change upon ligand binding, these motion types. And very briefly, this data captures how the same protein dynamically changes before and after it is bound to ligand, as illustrated here, or ligand. I get confused between that too. So if someone could talk to me about if it's ligand or ligand after the talk, I'd appreciate that. Now, this data set was more formally known as the Protein Structural Change Database, or the PSCDB, which contains approximately 1,000 pairs of structures. Each of these pairs of structures was labeled with one of 7 motion types that would later be used for our labels for classification. And after cleaning and processing the data, we arrived at 7 data quality thresholds, or data sets, where our highest quality threshold contained 143 pairs and our lowest data quality threshold had 402. And it's important to note that we actually dropped the other class, because in this particular motion type, there were not many proteins, and those proteins that actually were in it were not defined by a particular motion type, as the term other would suggest. We wanted actual defined motion types for these proteins. So ultimately, we were working with 6 motion types, or 6 motion type labels. So for each pair, we construct 3 PSN types, one of which is static and 2 of which are dynamic. And the most obvious model to use is obviously the static model, where given a pair, we construct a static PSN for the free protein structure and another static PSN for the bound protein structure. And we then extract network features from each of the static PSNs and compare them to see if there were any topological changes that occurred. So if there's a particular topological change that occurred, maybe we can link those to a particular motion type label. Then we extracted network features from each of the PSNs independently of each other and compared them only after the fact. So in this way, we are capturing the data dynamics implicitly. Is it switching? There we go. The other model was the dynamic PSN model, which is an obvious extension to the static PSN model, where given a pair, we construct a dynamic PSN for the free structure and another dynamic PSN for the bound structure, and we extract features from each of the PSNs independently and again compare them after the fact. Now, this is still capturing the data dynamics implicitly, but instead we're using our dynamic PSN model. And we call this the advanced dynamic PSN model for reasons for which I will explain very shortly. Now, from here, we ask, what can we do to capture these dynamics more explicitly rather than implicitly? So our third model addresses this, where given a pair, we construct a single dynamic PSN that captures the free and the bound structures simultaneously as different snapshots of the same PSN, such that when we're extracting features, we're actually extracting simultaneously across the snapshots, and hence capturing the dynamics explicitly. And this dynamic PSN model is described as being simple because it will only have 2 snapshots, as opposed to our advanced model, which can have many snapshots of varying amounts, depending on the size of the protein that we're working with. Now, what we expect is, because the only difference between the first model, our static model, and the second model is being that one of them is static and the other is dynamic, respectively, we're confident that the second model should outperform the first model. But with the second model having the advantage of being advanced, yet having implicit data dynamics, and the third model having the advantage of having explicit data dynamics, yet being simple, we're not certain exactly what would happen or what kind of performance we may get, but that's something that we'll see very soon. There we go. So for each of the PSN types, we extract corresponding graph features. And with these features and the 6 protein motion types that I described before, we apply them into a multi-label classification model. And for each of the 7 data sets that I also talked about earlier, we compute classification rate, where higher classification rate would indicate better performance. We also compare our method to 3 benchmarks. Firstly, the majority class, which is just the most frequently labeled class in a data set. We also compare to TM score and RMSD. Now, we did try to do a literature search to find other methods to compare to as well, but we ended up finding network-based and some physics-based methods, but for all these methods, we could not find any code. So given our methodology, we produce these results, where each of the colors correspond to a method, and on the x-axis are the 7 data sets that we test on, and on the y-axis is classification rate, where again, higher classification rate, better performance. Now, what you might find is that PSN-based methods consistently outperform the benchmark methods over all data sets. And between static and dynamic PSN methods, we actually find that dynamic PSN methods consistently outperform static PSNs, just as we had expected. But what's most surprising was between our advanced and simple dynamic PSN models, despite only having 2 snapshots in the simple PSN model, they were still performing within standard deviation to the advanced dynamic PSN model for the majority of these data sets. Which we believe that even though these simple dynamic PSNs do not have as many network snapshots as the advanced type, explicit data dynamics are seemingly very effective in classifying protein motion types, or ligand-bound protein motion types. So we're very happy to see that our methodologies are also robust to noise and data quality, as you can see. Now, ultimately, we would want to move towards explicit data dynamics, like the color purple, with advanced approaches like the color red, which means that we need more data, dynamic data for more than 2 intermediates. So just imagine if we had more intermediates. So overall, dynamic PSNs outperform static PSNs and the basic benchmarks in classifying protein motion types. I've shown that our current work addressed that dynamic PSNs work well on static data, and hopefully today I've shown you that dynamic PSNs are great on dynamic data as well. Our results on protein motion classification and other rigorous analyses will be made available on arXiv as well, hopefully in the near future. Still, we believe that if we had more data on intermediates, we can have more improvements, right? So in the future, we hope to test and improve the model with other dynamic data. Now, specifically, we thought of using molecular simulation data of protein folding pathways, but from what we can find, there are a lot of computational derived data, but no experimental data. And even for computational, there is no centralized data resource that's both adequately cleaned and labeled. Oh, excuse me. Didn't want to spoil that. We also thought of using co-translational protein folding data, but for both experimental and computational methods, there are no data. And unfortunately, perhaps you're thinking that AlphaFold could be the solution. But unfortunately, AlphaFold is not very good at predicting these 2 data types as described in this paper. And we'll be investigating in our ongoing work as well. So all in all, our dynamic PSNs seem to be really good at generalizing to dynamic data, and we certainly hope to test this model and design more advanced models on more newly available dynamic data. And before I end, I'd like to say, I would like to add that we discussed these kinds of challenges related to network-based analysis of protein folding dynamics, along with many other challenges in the field of network biology in our most recent perspectives paper, for which I'm a co-author of. Co-first author of. This was an amazing and an enormous effort by many authors, and we hope to shape the future of network biology with this kind of work. Thank you.
Thank you, Aydin. You have questions?
I don't seem to understand what these PSNs are.
Sure. Would you like
Is it like a simplified representation of the actual protein structure?
It's a
You
network.
have a node for an amino acid and an edge for an interaction.
So you're correct that nodes are amino acids, but edges are going to be derived if they are spatially close, if these amino acids are spatially close within the 3D fold.
Okay.
So that's how we draw edges.
So, okay. Thank you.
Hi, nice talk.
Thank you.
I actually had the same doubt, but now I think I understood better.
Sure.
In your final comparison, you compare against RMSD.
Mhm.
The problem with RMSD is the small changes in hinges, for example. They lead to huge changes in RMSD, so it's not a very fair comparison, I think.
Fair enough. Yeah.
There are other similarity scores like the Q that is used in protein folding from Peter Wodines and Josiane Chick that is similarly, to the fraction of native context compared to a reference structure, which could be whatever of your states. Have you explored using this to compare to your methods?
That was certainly an idea, and that's actually certainly in the back room. That's actually under process. So we're hopefully looking into that and seeing what other methods we can also compare to as well. One of the interesting things that we were trying to do was trying to find other methods as well, other published methods to test out, but unfortunately there was no code. But certainly that method, that is something that we can test on. So yeah.
Hi, good talk. Could you clarify how snapshots of a static structure represent dynamics?
So I might have misspoke. Let me go to that figure and hopefully it'll clear it up. So we actually have 2 different PSN types, one of which is static and one of which is dynamic. So, and the static PSN is essentially just one network that is describing just the native structure of the protein. The dynamic PSN is comprised of snapshots, and what the snapshots are trying to do are trying to capture a growing substructure and sequence in each of the snapshots. So you can think of it almost as an incremental growth in snapshots until the final snapshot, however many snapshots you'd like to use, you arrive at the native structure. So that would be the difference between the 2.
So does that represent some kind of physical process or?
Yes, yeah. So the physical process that we're trying to replicate is actually this kind of
Oh,
process.
got it. Okay.
Yep.
Thank you.
One question, how you define the edges? Which distance you use?
Yeah,
to-
so that actually is up to you. And our lab actually tested a lot of different ranges of drawing of designing edges, right? You can set a 10 angstrom length and say any two amino acids that are within 10 angstroms or lower, you can set an edge. We find that 6 angstroms is actually quite nice at designing our method. And in fact, in the previous paper, our 2022 paper, we've actually looked at why we've shown that using 6 angstrom length to design our edges was most effective for producing the classification results that we wanted. So,
Okay.
yeah.
Okay.
Hi, great talk. I'm no expert with the proteins,
Yeah, no
but
problem.
one of the networks you had was about ordered, static, and weight.
Yeah.
When you say weight, are you talking about the different interactions between amino acids, or is it how strong the interaction between amino acids?
Yeah, so weight actually, that's up to you how you want to define weight. For one thing, you can, like you said, you can define a weighted edge being how strong an interaction might be, or even distance, right? So maybe if they're very close, then you want a stronger weight, or if they're very far, then maybe a weaker weight. But yep.
Thank you.
Nice talk. Thank you. So you show the snapshot of the dynamic of the part, the snapshot 1 to 3 by the translating of the proteins. So have you actually put into the element of the kinetics of the translations? So the thing is like the codon usage is actually all kind of the differentiate.
Mhm.
So it means kind of some kind of the region is going to be the faster than the other. Sometimes it's kind of the stopped at some point, and then kind of the more network, more stabilized, and then just keep going on the translations. So
Yep.
have you actually considered those kind of the kinetics in your calculations?
Yeah, so I don't believe that I had considered that just yet. Actually, the way that we design our dynamic networks, we're trying to find a simple way to construct them at first. And certainly, I think this is a great starting point for our dynamic networks. But adding more kinetic information and particularly more biological information to this will be great. Yeah.
So you represent the structure in a somewhat simplified, more simple way. And then you show that you can recapture the classification of structures that is based on the structure that you're representing in a more simplified way, right? With the classification that you were talking in the beginning about.
Oh, in the beginning. Yeah. So
Then at the end, you're talking about conformational changes, and you're using the simplified structural representation, and you show that you can capture the changes that are there in the structure. To me, it seems a bit obvious that this dynamic thing that you have a smaller snapshot magnifies the signal that is only a small part of the movement, just like Gonzalo was suggesting that the RMSD is a measure that gets diluted for the entire structure.
Mhm.
When you have these partial things, you have less of a noise of areas that are not relevant. So I'm trying to understand what is the advantage of this. Why do you need this if you already have the structure?
could you define?
Why do you need the PSN if you already have the structure?
So if we take our PSNs and we recreate if we represent them as networks, for one thing, the features that we want to extract will be network based. There are multitudes of methods to extract features from networks. The other thing I think that I want to address, which I think is the first part of your question, is dynamic PSNs were somewhat of a novelty when they were first invented. And so we wanted to try to see how do they work with static data in the task of protein structure classification. And hopefully I've shown to you that it's been effective. Even though it might be obvious that that a certain construction of the dynamic PSN would be more effective than others. What we really wanted to show in this current work was that this model is generalizable to dynamic data. And we needed to show that by performing this kind of experiment. So in reality, what we're trying to really show is that these dynamic PSNs are excellent, not just on the static data, which was our previous work, but also are great for dynamic data purposes. And because they're quite simple and not as highly detailed as the 3D structures, well, for one, I believe I would have to confirm it, but I would imagine that they would be faster than just running the 3D structures themselves. And obviously, I'm very biased towards network-based analysis. So I believe that the network features that we extract are quite important, and hopefully we can use these for other dynamic processes. Hopefully I answer your question.